Good morning, everybody. Welcome to Life Sciences Grade 12. My name is Duduzile Kupeka, and we are continuing with the topic of assessment memorandum. So we are doing the memo. That is my email address. If you have any more questions about the assessment, you can find the assessment online, and I can email the memo to you if you are struggling. Anyway, question 1.6. A normal healthy person was placed in a cold room. After 30 minutes, the thermal image shown below was produced. This thermal image indicates the temperature of different parts of the body. Lighter colors on the scan indicate temperatures lower than normal body temperature. So this person here, meaning the white, that's the lowest temperature in this person's body. Then the gray is lower than the black, but higher than the white. And then the black is the normal body temperature, because we are told the lighter colors indicate lower temperatures. Question 1.6.1. Which color, black, gray, or white, represents the normal body temperature? And we come to the image again, and we read the information provided to us, right? Lighter colors on the scan indicate temperatures lower than normal body temperature. So meaning the normal body temperature is the black. The lighter ones indicate lower temperatures. This one is the one that is normal body temperature, the black. 1.6.2. State what occurred in each of the following parts in this person's skin during temperature regulation. A, what happened with the blood vessels? And B, with the sweat glands. So we are focused on this person's skin during temperature regulation, right? The skin. So let's look at the skin. The skin is white, meaning it is colder than all the other parts of the body. So this means, yeah, this person is actually feeling cold, né? Because it's the skin and the inner parts of the skin. It's cold, it's cooler, it's lower than normal body temperature. So what happens during thermoregulation? When the body is feeling cold, it needs to retain as much heat as possible. So what would happen? The blood vessels on that are on the skin, imagine they are coming from the dermis to the epidermis. They will be constricted. Why? So that it prevents the flow of blood to the surface of the skin. Because if blood were to flow to the surface of the skin, blood is hot. It's carrying heat energy. So it's carrying heat. If it were to go to the surface, that heat would then evaporate outside the body. But now, because this body is trying to retain as much heat as possible, it will constrict the blood vessels which are towards the skin, to ensure that the blood keeps on flowing to the internal parts of the body and that no heat is lost. So what happens in the blood vessels? The blood vessels will be constricted, or through that process we discussed called vasoconstriction, which will occur. So that's how you say it. You'd say the blood vessels constricted or vasoconstriction occurred. And then what happens, and then what happens with this? What also happens after vasoconstriction? Less blood will flow in the blood vessels. And then B, the sweat glands. What happens with the sweat glands? You remember sweat glands will also be constricted. Why? They only relax, or they are dilated rather, when it's too much heat, so that they allow the flow of water outside the body. So the flow of water, it will flow, the water that will be carried out will flow, and then it will be released as sweat, whereby it will evaporate from your skin when you are feeling too hot and your body is trying to cool down. But now you are not feeling too hot. What are you trying to do? You are feeling cold. Your body is trying to retain enough heat. So, I'm sorry, guys. So what will happen? The sweat glands will become less active and less sweat will be secreted, because the body only releases sweat when it needs to cool down. And then 1.6.3. not only, right, when we're discussing another topic of thermoregulation, that's all I'm saying right now is referring to thermoregulation, nothing else. 1.6.3, name the part of the brain that is responsible for thermoregulation. Which part of the brain? It is called the hypothalamus. That is where you see it functions with a lot of other glands. This one, the hypothalamus. It's the part of the brain responsible for thermoregulation. Then 1.6.4, state one way in which the thermal image would differ if the person was placed in a hot room for 30 minutes. So if a person was placed in a hot room for 30 minutes, this whole body will appear black or black and gray, and there will be no white areas. Why? Because the skin also will be very, very hot due to the temperatures in the room. It would be feeling hot. So the whole body will indicate either black, which is the normal body temperature, or black and gray, meaning some parts would be just a little less hot than the others. But there wouldn't be any white, because white indicates the very, very low temperature. So that was topic on thermoregulation. If you are struggling a bit to follow there, go review the videos we have. We taught the topic thermoregulation this term, and you can be able to get up to speed. And then section B, question 2, 2.1. The diagram below represents a phase of meiosis. Oh, guys, I think I flew. So it represents a topic of a phase of meiosis. You see here, you have two cells, A and B. Identify this phase of meiosis here. Which phase is this? Remember, it is telophase 1. And then 2 point, you see, you indicate with these two cells over here. There are two cells over there, and the chromosomes are still intact. You have the chromosomes, you have two pairs of chromosomes in each of the cells. Draw a diagram to show only the two gametes formed from cell A. No labels required. So we are following from cell A. You'll draw a diagram to show the two gametes that will form from cell A. There. You can either indicate it like this or like this. Basically, you see this one. It's basically the same thing. Look, let's look. It's very strict on cell A. So you need to look carefully with cell A. You see you have the whole black, and the other one is black and a little white. Then the other one has black, white, black. Then it has a white. So when you draw it, you need to indicate that has happened. Remember, after the cell divides, then which part will move with each gamete. So this is how you would indicate it. Initially, you had how many pairs? You had two pairs of chromosomes in cell A. Now, you would have one pair chromosome each. So it's either this one on the left or this one on the right. If you indicate any one of them, you would be correct. And then question, there are no labels required, so you can just draw it and leave it at that. 2.1.3, tabulate two differences between prophase 1 and prophase 2. So here, you draw your table. You draw your table, you label prophase 1, you're labeling prophase 2. Then you write the differences. For prophase 1, the cell is diploid or it has homologous chromosomes. But in prophase 2, the cell is haploid and does not have homologous chromosomes. So you are comparing, right? You're writing the differences. So that's why it needs to match. When you're speaking about the diploid cell, or you see, then you need to also compare the same thing in prophase 2. You're comparing, you're not just writing facts about them, you are comparing between prophase 1 and prophase 2. The second one is crossing over takes place in prophase 1, but in prophase 2, there is no crossing over that takes place. Now you have written two differences, right? 1, 2, 3, 4. You get a mark for each difference you note down. Then you'd get another mark for what? For drawing the table. That's why I always emphasize, even if you don't know the answer, just draw the table, label it accordingly, and then you'll write down the differences at a later part, but you get a mark for just drawing the table. And that really doesn't require much. You know how to draw a table. So you draw a table, you will guarantee the mark. Then you can start putting down the differences, right? And also, as you note here, we mark only the first 2 questions. So if you have written maybe 5 differences each side, and then you happen to write the wrong ones in the first, if your point 1 and 2 are wrong, but your 3, 4, and 5 are correct, unfortunately for you, it won't be marked, and we wouldn't even know, because what we do is only marking the first 2. As the question is clear, tabulate 2 differences. So make sure the ones you write are correct, and make sure you follow the instructions. The ones you are most sure about, write those ones. And it's easier for you if you're asked only for 2 differences. Then you can work through that easily. Ne? And then 2.2. A clinostat is a device used to investigate plant growth responses. It has a disc that rotates very slowly when the clinostat is switched on. During an investigation on plant responses to light, the procedure below was followed. Remember, we did this work again? And then this was the procedure that was followed. 3 pot plants of the same species were used. Each pot plant was placed on one of 3 identical clinostats. Each set of apparatus A, B, and C was placed in a box with a single opening. And then each clinostat was treated in a different way over a period of 5 weeks. So each clinostat was treated in a different way over a period of 5 weeks, right? Remember I showed you a video of a clinostat in one of the lessons we did here. And while they're trying to test for here, a plant's responses to light, which is called what? Phototropism, remember? So 2.2.1 now, based on that setup, that experiment, name the plant growth response to light. It is phototropism. That is plant growth that is responding to light. It's called phototropism. And then 2.2.2, state 2 factors that were kept constant during this investigation. The factors that are kept constant were what? The same species of plants was used in each setup. Identical clinostats were used in each setup. The same period of time, which is like 5 weeks each, was used for each setup. Each apparatus was placed in a box with a single opening. The opening on each box was in the same position, or was the same size, or was allowed the same amount of light to enter. So the question is asking only for 2 factors. So if you can write any of these 2 here, you'll be correct. Any of the 2. If again, the same rules apply. If you write more than 2, the only ones that will be marked are the first 2 answers. And then that is it. As you see, all that information I found here, it's nothing I came up with. All of these, it's stated here that same species of pot plants were used. Each pot plant was placed on one of 3 identical clinostats. You see, all of these points are listed here. So we got all the answers on the information given. That is why it's important to read and understand information given before you start answering. Excuse me. And then 2.2.3. Give 1 reason why the results of this investigation may be considered to be unreasonable. Only 1 reason. Why? The investigation was only done once, and it was not repeated. So that could be the reason. Remember when we learned about this topic and we did this, I showed you the video of the experiment, and we also went through the whole write-up of the scientific report. What needs to be done? Even when you are done doing your experiment, you need to repeat it to test the reliability of your results. You need to repeat it, just again, repeat it, even if you repeat it maybe 3 times, to be sure that you will get the same results. And then the other point could be only 1 plant was used in each setup, meaning the sample size was too small. You'd find that because of this plant species, it would react that way, but a different plant species would react differently. So any one of these 2 points, you would be correct, because yes, they show these ones, these are valid reasons as to why these results might not be reliable. And then 2.2.4, in which apparatus, A, B, or C, was the clinostat? A, in which one was it switched on and rotating slowly? And in which one was it switched off but manually rotated through 180 degrees once a week? Which one? A, the one that was switched on and rotating slowly, is B. Let's go look at it. Switched on and rotating slowly. You see this one, it was switched on but rotating slowly. Why? The plant grew upright, meaning it wasn't confused about the light. Remember, the shoots will go up following the light and the roots will go down for moving further away from the light. And here it was switched on but rotating slowly. And we see based on how the plant grew. It grew it was rotating slowly. Okay, noted. It was rotating quite slowly, right? So it was rotating quite slowly, and you see it moved straight up, just up there. And then the next one here, question B, it was which one was switched off but manually rotated through 180 degrees. This one was switched off, but it manually rotated through 180 degrees a week. It is plant A. Sorry, let's go back to plant A. There. It was switched off, but manually rotated to 180 degrees once a week. You see that at one point it was up here, then it changed direction, then it was moving towards the light. Then again, it happened that it was switched off, then it moved away. And then again, you see with the spiral shape of how it happened. Then this one here is the normal one, the normal conditions in which the plant grows. Why? As you can see, the shoot, it bended there to follow the light. There was a bend to show that it's following the light. Exactly like that. This one was so confused, it never followed the light. This one, at some point when it was switched off, it would follow the light. When it was switched on again, it would move away, confused. Switched on, switched off, follows the light. Switched on, again, confusion. This one was never switched off. It was kept on rotating slowly. That's why it just moved up. And then this one over here, it moved in the normal way, whereby we know and we learned the auxins here produced in the shoots would allow the elongation and the bending to happen, whereby the plant or the shoot will follow the light. And then question 2.2.5, explain the effect of the unilateral light on the distribution of auxins in the plant in apparatus C. Like I've just mentioned to you, apparatus C, we're explaining the effect of auxins here. What is the effect of auxins? It's the normal one. The way we learned it is that the auxins moved away from the light or were destroyed by the light. Then what happens? So that the darker side had a higher concentration of auxins, and the lighted side had a low concentration of auxins. So that's what happens. Remember, auxins will move away to go to that, so that the darker side will have a high concentration of auxins. Then the process of elongation happens. Then the lighter side has a low concentration, it will bend. Then the shoot will face the light, the bending there. That bending there is because of this effect of auxins, this bending here. This here, you could see it happened, but as soon as it was switched back on, then it continued the confusion. This is the revision of, this is covered in the topic of plant responses. And then this one was particularly dealing with phototropism. So guys, you can still, we have that lesson in the YouTube videos. Go through, search by that title, you will find it there. And in the textbooks as well. Remember, I always advise to go through different textbooks to cover your material. Okay, and then 2.3. The graph below shows the speed at which impulses are transmitted along sensory neuron axons of increasing diameter when the axons are myelinated, which means they are covered with myelin sheath, and unmyelinated, which means there's no myelin sheath present. So what are we looking at? We're looking at the speed at which impulses are transported along sensory neuron axons. So this is the graph we have. The topic: comparison of the speed of impulse transmission in myelinated and unmyelinated sensory neurons of increasing axon diameter. On your x-axis, you have your axon diameter, which is in micrometers, and then you have the speed of impulse, which is in meters per second. The line with the dashes there, the dashes here show you myelinated, and the straight line shows the unmyelinated. This line here, with no spaces in between. So 2.3.1, describe the direction of the impulse with a neuron. The direction of the impulse with the neuron. The direction of the impulse with the neuron, it would move from the dendrite to the axon. Remember that it moves from the dendrite to the axon. And then 2.3.2, give the diameter range in micrometers when the speed of the impulse is faster in unmyelinated axon than in myelinated axons. So we are giving the diameter range. Range is very important here. In micrometers, right? The diameter is given in micrometers anyway. When the speed of this impulse is faster in unmyelinated axons than in the myelinated axons. So we are comparing here the unmyelinated axons and the myelinated axons. So you write the part whereby the unmyelinated axons, the speed of this impulse was faster in the unmyelinated than it was in the myelinated. And we're looking at our graph here. You can see that from 0 to 1, or 0 to 0.9, because from 1 you can clearly see, it's not very clear. So you can say from 0 to 1 or 0 to 0.9, this part over here. Unmyelinated, the speed of the impulse is faster than the speed of the myelinated axons here. Then question 2.3.3, describe the relationship between axon diameter and the speed of the impulse in myelinated axons. So let's describe that. The relationship between axon diameter, if you are the graph here, that's what's indicated on the x-axis, and the speed of the impulse in myelinated axons. So we are only focused on this line, which has spaces. We're looking at the relationship there. What is the relationship? As the axon diameter increases, the speed of the impulse also increases. That you can see, it's a constant increase. The more the axon diameter is increasing, the higher up, the more the speed also increases. Or as the axon diameter decreases, the speed of the impulse decreases. So when you're looking at from here, 0 to up there, you see, you would speak of the increase. But if you start from the 4, the side, on the right-hand side to the left-hand side, then, or from up and down, then you are noticing a decrease. So either way, they mean the same thing. It means when the diameter decreases, the speed also decreases, or when the diameter increases, the speed also increases. That is 2.3.3. And then 2.3.4, which is the one we haven't answered. Use evidence from the graph to explain the effect of multiple sclerosis on a sufferer whose motor neuron axons are greater than 1 micrometers in diameter. So we're studying what? The effect of multiple sclerosis On a sufferer whose motor neuron axons, the motor neuron axons of this person are greater than 1 micrometers in diameter, the speed of the impulse will decrease, right? Resulting in it taking longer for impulses to reach the effectors, and then the person will react more slowly. You see that? Whose person is it greater than, it is greater than 1 micrometer, meaning that one there is, it shows the effect here of this unmyelinated. So that person with multiple sclerosis, it would react this way, like the unmyelinated axons over here. That's how the reaction would happen. And you see, from if it's greater than 1 micrometers, the axon diameter, then it moves quite slowly. The speed has decreased. So that's why we are comparing these. Understood? And then 2.4, we have 2 types of hearing loss occurring in humans. Yeah? There are 2 types. There is conductive hearing loss, which occurs when sound vibrations cannot be conducted through the outer and middle ear. And then the sensorineural hearing loss, which occurs when sound waves in the inner ear are not converted into nerve impulses, or when the impulses cannot be transmitted to the brain. So it can be due to the conductive nature of the ear, or the senses. It's that the senses are affected in the neural region here. So this diagram below represents the human ear. Remember the human ear, you did it in tab 2. This is the human ear over here. 2.4.1, give the letter and name of the part that A, which transmits impulses to the brain, and which one that allows pressure to equalize between the outer ear and the middle ear. Which part of the brain of the ear transmits impulses to the brain? It is the auditory nerve. Auditory nerve is part F over here. This here, part F, the auditory nerve, that is the one that transmits impulses to the brain. And B, which one allows pressure to equalize between the outer ear and the middle ear. That is the eustachian tube, which is G. Eustachian tube allows pressure to equalize between the outer ear and the middle ear. There, this is the eustachian tube. You can even see with the tubes. Question 2.4.2. Give only the letter of 2 structures in the diagram of the ear that, when damaged, would result in the following. A, conductive hearing loss, and B, sensorineural hearing loss. Remember the 2 types of hearing losses we studied. So you need to give only the letter of 2 structures in the diagram. It's even written in capital letters. Letter of 2 structures in the diagram of the ear that show that when damaged, so which part in the ear when damaged would result in these 2 conditions. Conductive hearing loss would be because of B and C. Let's see B and C. There, if this is here, B and C, it will result in conductive hearing loss. Why? Because this is when here the sound vibrations cannot be conducted, and these parts are responsible for the conduction of sound vibrations through the outer and middle ear. Remember? So you look at their location and you also look at their functioning. So if these 2 were damaged, then you'd have conductive hearing loss. And then E and F would result in, where is E and F? There is E and F, that part, that region there, E and F. That would then result in sensorineural hearing loss, because here the sound waves in the inner ear wouldn't be converted into nerve impulses, you see? Or they wouldn't be transmitted to the nerve impulses, wouldn't be transmitted to the brain. And then next question, 2.4.3. Middle ear infections are a common cause of hearing loss. State one way in which middle ear infections are treated. How do people treat middle ear infections? Grommets will be inserted in the tympanic membrane. or what? Antibiotics. So both of these two, depending on how severe it is, you can either insert grommets in the tympanic membrane, or the person can be given antibiotics to treat the infection. Then they'll recover. And then 2.4.4, name the part of the ear where earwax is produced. It's on the auditory canal. That's where the earwax is produced. Auditory canal. And then 2.4.5, explain why hearing loss due to earwax is usually temporary. Why is hearing loss due to earwax usually temporary? Why? The earwax can be removed. Of course, the earwax can be removed from the auditory canal. This will then allow sound to reach the tympanic membrane, which will also allow the tympanic membrane to vibrate freely. So that's when it's temporary. Earwax can easily be removed, right? But the correct ways of removing it. So that is the effect of it. If there's too much earwax, then it could cause temporary hearing loss. But if it's removed, then it will allow the sound then to reach the tympanic membrane, allowing the tympanic membrane to vibrate freely, because when it's clogged with the wax, it can't vibrate freely. And then question the diagram below represents some parts of the male reproductive system. This is the male reproductive system now. Then we need to identify parts A, B, and C. What is part A? What is part B? What is part C? A is the testis. You see, there is the testis there. This ball over here. B is the epididymis. Epididymis over here. That is our structure B. And then C, what is C? There's a scrotum. So that is the bag or the sac that is carrying all of this. It's enclosing all of this. This is the testis, epididymis, and then the scrotum over here. And then 3.1.2, describe the process of spermatogenesis in part A. Part A is in the testis. So you're explaining the process of spermatogenesis in the testis. Remember, so these are linked. If you didn't know exactly what A is, you might struggle to answer this question. So what is the process there? Under the influence of testosterone, the diploid cells or the germinal epithelium in the seminiferous tubules of the testis will undergo meiosis to form the haploid sperm. I wrote these in point form so that you can follow easily and you can take note of what is important to write. So it happens, it's important you mentioned that it happens under the influence of testosterone, which is that hormone responsible for the production of testes. So in the influence of in the formation of sperm cells, right, and various other functions in the male reproductive system. So it's under the influence of testosterone, whereby the diploid cells in the seminiferous tubules of the testis will undergo meiosis to form the haploid sperm. And remember why it has to be haploid, because sperm cells, the reproductive cells, which is the sperm cells in males and egg cells in females, are transferred in their haploid forms. Why? So that they can only pass on 23 chromosomes. So it will be 23 from the sperm cell, 23 from the egg cell, and then when they fuse together, it will be 46 chromosomes for the resulting child, or the resulting eggs, or the resulting fetus rather. Ne? So it's 23 from the, what's this, 23 from the sperm, 23 from the egg. It's unlike, this one functions differently from all the others because all the other cells in the body function differently, right? They function differently in that, what do they do? All the others in the body function differently in that they are always in their 46 chromosomes or 23 pairs state, except for the reproductive cells, which are the egg cells and the sperm cells. So that is the process that happens during spermatogenesis in part A. And then 3.1.3, test results show that a man has a low sperm count. Explain why a doctor would advise the man to wear underwear that is not tight. So let's state here, these test results, this man here, even with all the processes going away, well, it shows that this man has a low sperm count. So why then would a doctor advise this person to wear underwear that is not tight? Why? What's the point of underwear that is not tight? The testis will be away from the body. The temperature of the testis will therefore be lower than body temperature, or there'll be less pressure on the testis. And this is playing a role in successful sperm production. Because when you're wearing tight underwear, you are bringing your testis closer to the body. And then the heat will then move from the body and it will affect your testis. So your testis will have the body temperature. And they are not supposed to have body temperature. They are supposed to have a lower temperature than the rest of the body, because sperm production happens effectively under low temperatures. But in high temperatures, then sperm production is affected. Another way you can phrase this is that tight underwear will pull the testis close to the body. The temperature of the testis will be too high or higher pressure on the testis. The sperm will not mature, or sperm production is negatively affected. Either way, as long as you make it a point that it comes across that it is because of the testis being closer to the body that it will get the body's temperature, which is not supposed to. It needs to have a cooler temperature. So if you move it away from the body, it will get to hang and be cooler. And then what? There's no pressure there as well. And then that will then ensure that the process of sperm production is not affected negatively, as the sperm will now be able to mature. But with the tight underwear and the hot body temperature, then the sperm is not maturing or its production is affected negatively. And then 3.1.4, during a vasectomy, the vas deferens from both testes is cut out. This is the vas deferens. You see that tube there. The vas deferens from both testes is cut out. Explain one reason why a man who A, does not want to have children will choose to have a vasectomy, or B, has had a vasectomy and is still capable of ejaculation. So let's deal with A first. What reason would be there that a person who doesn't want to have children will choose to have a vasectomy? Why? Why would a person who doesn't want to have children choose to have a vasectomy? Because when you have a vasectomy, there will be no sperm in the semen, right? Therefore, no fertilization can take place because you don't want to have children. So if you cut off this tube here, this tube, it means you will be cutting off the supply, the sperm that can flow. Then the sperm can't be transported there. So the semen will just travel empty and there will be no sperm in the semen. And what is responsible for reproduction or fertilization? It is the sperm. And then B, which is dealing with the topic here, this man has had a vasectomy but is still capable of ejaculation. Why? The fluid part of the semen will still be produced by the accessory gland or the seminal vesicles or the prostate gland, or also corpus glands. So all these glands will still produce semen. But the difference between this semen is that the semen now has no sperm, as we've stated here in A. There will be no sperm in the semen. So the semen will still be there, except it will be missing the part that is required for fertilization, which is the sperm. So yes, this person will still be capable of ejaculation, but they will not be transmitting any sperm because the sperm is not being transported. You see, this tube, if it was removed or cut, then the sperm can't pass through. The sperm that was here in the testis cannot pass through now. That is the reason why. And then that is it for 3.1. 3.2 now. Maize was genetically modified to increase its resistance to insects. This GM maize was introduced into a country to increase food security. So genetically modified maize was introduced to this country to increase food security. Why? How was it modified? So that it has increased a resistance to insects. Has increased resistance to insects. The graph below indicates the number of maize farmers in the country that were diagnosed with severe depression over a period of 10 years. It also shows the area of agricultural land that was planted with GM maize crop. So let's study the graph here. On the x-axis, you have the year. On the y, you have 2y. We have on the left y, you have the number of farmers with depression. On the right y, you have area planted with GM maize, which is times 1,000 hectares, right? This is the heading of the graph is number of farmers diagnosed with depression and the number of hectares planted with genetically modified maize from 1997 to 2006. So these are the years indicated here. So let's study the graph, the years. 2000, we have, that's when we start to see the area planted with GM maize. When we study the year and the area planted, we'll focus on the right y-axis. If you're focusing on the number of farmers with depression, you'll focus on the other side. So you'll see from 2000 up until 2006, the area that was planted with GM maize was increasing, right? As you can see, in 2000, there was like 0, then it moved, it increased. In 2006, you have 500. It's 5,000 times 1,000, so it's like 500,000 hectares, which are planted with GM maize, because it's that 500 times 1,000, right? Are you following? Because that's what is written there. So it's 500,000 hectares now that are planted with GM maize. So it shows you the increase over here. And then when you look on our left side, you see cases of depression. The cases of depression were first recorded in 1997. Then it decreased a bit in 1998. It kept on, and then it increased. You see, it fluctuates. It was highest there at about 2000, the cases of depression. Then they went on and they were decreasing now at a slow rate, but they decreased. You see, kept on decreasing the number of cases of farmers with depression over the years. Okay, firstly, before we start, there's the question 3.2.1. Give a definition for food security. What is food security? Food security is defining a term whereby all people should have enough food. The food should be nutritious or of good quality and must be accessible at all times. That is food security. When we speak of food security, we are saying all people have access to food. And that food they have access to is nutritious and is of good quality and is accessible at all times. 3.2.2, name 2 farming practices other than growing GM foods that the farmers may use to improve food security. Besides genetically modifying the food, they can use fertilizers, they can use monoculture, pesticides, improve the irrigation, crop rotation, and artificial selection. All of those processes could assist in improving food security. 3.2.3, from the graph, state how many farmers suffered from depression in 2003. So we go to the graph, look at 2003. There, you move up. I'm not drawing very straight, but there. 2003, number of farmers with depression. Move over here. It's 2,500, right? 2,500 farmers suffered from depression in 2003. I hope you can see how I followed the graph. 2003, that's the line there. with farmers. Be sure you're looking at the correct y-axis. The one here is asking about the number of farmers with depression. So we are focused on that one. If it was asking about the area planted with GM crops, we were going to go to the right. But now we're looking at farmers with depression. There. And then 3.2.4, explain why more farmers suffered from depression before the introduction of GM maize. Why? Why did they suffer from depression before the introduction of GM maize? Crops were destroyed by insects. This led to a lower yield or added expenses for insecticides, and therefore resulted in lower profits. So this loss that they were incurring, because these are their livelihoods, these are their businesses, they affected their well-being. And that's why they had started suffering from depression long before GM maize was introduced. You understand? So it means all the other methods they're using were not really working effectively. That is why then they suffered major losses due to the crops being destroyed, and then they are losing their profits because of the lower yield. I hope that is clear. Okay, guys, we are out of time now. On Monday, I'll do it because it's only the last. Okay, or what? I don't know. We'll see. I'll see you next week, but we have to continue with the topic next week. So maybe on Monday, briefly, the first 15 minutes, we'll cover the last part of the question paper, and then we'll continue with our work. Okay, guys, have yourselves a great day, a great weekend. Take care of yourselves. Bye.